The human immune system is vital and saves us from certain death by infection. It has two types of responses, innate and adaptive. The innate responses form the first line of defense against invading pathogens through direct defense mechanisms such as protective barriers. It also activates the adaptive immune system, which has two types of responses, cell-mediated responses, which use T cells, and humoral immune responses, which use antibodies produced by B cells. B and T cell mediated functions ultimately lead to pathogenic destruction. In this presentation, we will look at how antibodies are generated, how they function, and how they can be used to treat diseases such as cancer. Just briefly, it is important to know where antibodies come from. Like I said, antibodies are produced by a type of white blood cell called B lymphocytes. These are also called B cells, and they are produced and develop in the thymus and bone marrow. These cells then migrate to peripheral lymphoid organs, such as the spleen and lymph nodes, where they can then react with foreign invaders. So what exactly does an antibody look like? An antibody is essentially Y-shaped and is made up of 4 chains. The tips of the Y are antigen binding sites. An antigen is something that stimulates an immune response and is usually a pathogen. Antigens have small regions called antigenic determinants, and each antigen may have several of these. These are what the antibody recognizes and binds. Another important feature is the stem of the Y, called the Fc region, and this acts as a marker for destruction by binding to other immune cells with Fc receptors. The humoral immune system is responsible for the generation of antibodies specific for each antigen. As an antigen may have several different antigenic determinants, a normal immune response would therefore require a mixture of antibodies that can recognize all of these determinants. These are called polyclonal antibodies. If we have a look at how polyclonal antibodies are produced, this might make a bit more sense. If we inject an antigen into the spleen of a mouse, it will mount an immune response and produce a number of B cells. These B cells will produce antibodies which are specific for each antigenic determinant. If we took the blood serum from this mouse, we would find a mixture of antibodies that come from various B cell clones, and these are the polyclonal antibodies. Not so long ago, scientists found a way to produce antibodies that are derived from a single B cell and are specific for one determinant. These are called monoclonal antibodies, and these have become very useful therapeutically in recent years. To produce monoclonal antibodies, we again inject an antigen into a mouse, except this time we remove the B cells and then fuse these with mouse myeloma cells. This produces hybridoma cells which contain parts of both of these cells. Myeloma cells are able to multiply indefinitely, and so these hybridoma cells are then able to produce an infinite number of antibodies. If we isolate each clone and allow them to multiply, they will give rise to monoclonal antibodies that recognize a single type of antigenic site. We can then look at how monoclonal antibodies would react to a foreign invader. So we can see how each monoclonal antibody is specific for only one antigenic determinant. As opposed to polyclonal antibodies, monoclonal antibodies are advantageous therapeutically as they can be continuously generated with high specificity, with which they bind to their target and induce cell destruction. In recent years, monoclonal antibodies have become one of the largest classes of new drugs approved for cancer treatment, and due to their effectiveness, some cancers are now considered to be chronic diseases. One example in particular is in advanced metastatic breast cancer. In about 20 to 30% of breast cancers, the tumors overexpress a receptor called human epidermal growth factor 2. This is also called HER2 for short. A monoclonal antibody called trastuzumab was produced to target these receptors. Normally, when stimulated, two HER2 receptors will need to come together and dimerize to send activating signals inside the cell. But when trastuzumab binds to HER2, it prevents this dimer formation and therefore prevents HER2-activated intracellular pathways. These pathways normally increase cell survival, cell cycle progression, cell proliferation, and cell motility. but by blocking these pathways, trastuzumab effectively reduces tumor size, progression times, and improves survival rates of the patients. This is just one example of how monoclonal antibodies are used to treat disease. However, there are currently 35 other approved monoclonal antibodies and about 250 more in production stages. Thank you for listening, and I hope you learned something.